Hello, hello, hello. How's everybody doing today? Welcome to class. First exam is over. Is everybody excited? I've got my coffee right here. Horrible coffee it is. I think I need to. I'm doing one of those K-cups here in the teacher's lounge, and it's awful. K-cups aren't good to start with, and I think this one has been sitting around since March or April, so it's a little bit challenging to drink. Hi, Gita. Good to see you. Lydia, good to see you. Nicole Ortiz. Erica Schultz. Erica Schultz? You're in my developmental class, Erica. What are you doing here? You're more than welcome to visit, but I thought you were my developmental student. How are you doing? Ash Guff. Nicole, fantastic. Yeah, you're a couple hours early, Erica. Not bad, though. I appreciate your motivation. Hello. I saw it pop up. You're in the wrong class. Hello, Hannah. How are you doing, folks? I've actually invited a friend of mine, a Dr. Fitzgerald, the clinical psychologist who you met the first week of class. I'm not sure if he's going to be able to make it today, but I invited him to come see our broadcast. Eric is my kooky, crazy developmental psych student. She took intro with me last semester, and you just never know what that crazy Eric is going to do anytime. Hello, Maya. It's good to see you. Wow, got a pretty good crowd. It's about 16 people here today. 10:59, so we'll maybe wait one more minute. Who's drinking coffee? Anybody drinking coffee? Give me a coffee check-in. Actually, let me slide over here just a little bit. Get my camera. Me, there you go. Nicole, Etta, Rachel, there you go. All the coffee nuts just like me. Fantastic, fantastic. Wow. Okay, so it's 11 o'clock. Before we get started, let me see. The first exam was completed this week. You should have done your homework. You should have done the discussion board, and also we had week four of the neuromatrix. So let's go ahead and take a quick update on the neuromatrix.
Welcome to the neuromatrix.
Welcome to the Neuromatrix Update, Week 4. This week's Neuromatrix contest was all about the developmental theories of Erik Erikson and Jean Piaget. How would our Neuromatrix competitors handle the pressure of these developmental giants? Okay, let's stop that then. Clearly that's not going to work. Hold on just a second, folks. Okay. Looks like it. Okay, good enough. Well, fantastic. So we couldn't do the update this week. I'm sorry, the CPU usage is a little bit too high. Hope everybody's doing well this week. Welcome to class. Welcome to class. So this week we're going to be talking about vision. Actually, we're going to be talking about sensation and perception. Today we're going to be talking about vision. So what we're going to cover today, we're going to cover the basics of sensation and perception. We're going to talk about the location of the sensory cortices. We're going to talk about the visual pathways. We're going to talk a little bit about color vision, and we're going to talk something about sensation and perception today. Okay? How's my video stream? Okay. Yeah, sometimes when I try to incorporate the media files in here, it works earlier in the morning, but I'm not sure what happens between 9 o'clock and 11 o'clock. But for some reason, it just doesn't seem to work as well. Holy cow, what language are you speaking, Edita? Is that Polish that you're writing? Pretty impressive, Maya. Maya, you speak Polish as well? That's pretty interesting. Go ahead and type in the chat bar and let me know. Polish. Does Maya speak Polish as well? That's pretty impressive. Wow. All right. I hope you're talking about class. Nice. You're from Poland as well, Maya? Awesome. Fantastic. Well, yeah, we've got 35 countries represented in this class. Isn't that amazing? We have people from 35 different countries around the world. We've got 5 continents. Yep, 5 continents represented in this class. Pretty stinking impressive, huh? So let's go ahead and get started talking about sensation and perception. Now, what I want you to understand is that there are two processes to you experiencing all these senses that you experience. The first is sensation, and the second is perception. Now, you can think of sensation as recording the information into your neural content, into your neural tissue, right? So sensation is the lines that you hear, the colors that you hear, the pitches that you hear, all the different feelings that you experience. Those are all what we would call sensations. Now, in order for you to get the full sensory experience, you have to be able to understand what it is you're looking at. And that's called perception. So sensation is getting the information into your neural material via action potentials, and perception is you understanding what you're looking at. Now, the first part of this process is going to be receptor cells. If you'll remember from chapter 2, we talked about sensory neurons and motor neurons. And I told you that sensory neurons carry information from the periphery of your body to your brain. And then the motor neurons are carry information from your brain to your body parts to move them. Okay? Now, that's you have 2 types of neurons, but there's actually a third class of neurons we'll call receptor cells. And the job of receptor cells is to turn physical energy into patterns of neural impulses. We're going to call the process of turning physical energy into neural energy, we'll call that transduction. So if you're feeling your arm right now, when you squeeze your arm, you have receptor neurons that record that pressure as patterns of action potentials. When you look at a picture and the light comes in your eyes, you have rods and cone cells who turn that light information into patterns of action potentials. We're going to call that process transduction. And that's the process of recording physical energy into electrical energy. All right. And so we have all kinds of receptor cells. We're going to talk about rods and cones today. Those are the receptor cells in your eyes that turn light energy into patterns of action potentials. We're going to talk about hair cells tomorrow, and those are the cells in your inner ear that turn wave energy into patterns of electrical energy. We're going to talk about taste receptors or taste buds, and they turn chemical energy into patterns of electrical impulses or action potentials. And we're going to talk about olfactory receptor cells, which also, they're located in your nose, and they turn chemical energy into electrical energy action potentials as well. And you have a host of different sensory receptors that record pain, pressure, temperature, and itch into patterns of action potentials. So what I want you to do is to understand the difference between sensation, which is recording action potentials, and then if you'll remember last week, we were talking about consciousness, that video from Anil Seth. You turn the things you see into things that you can perceive and understand. You know what? I can actually give you a good example of that. You folks may not know this, but learning your shapes is a very, very important pre-writing skill. And you use the general shapes around letters to help you figure out what words you are reading. So if you would, I'd like to go to my whiteboard real quick and see if I can demonstrate this to you. You should be looking at my whiteboard right now. Do you see how I have dog and cat written here? And I have boxes written around each of them. Does that make sense? So you see how the dog has the tall box, then the square box, and then the G hanging down low? I want to see if you can guess what animal I am writing right now. If you would, try to guess and tell me what animal you see me writing. Based on the boxes, what word did I just write? Can you tell me what animal I just wrote on this board? Holy cow, elephant. How did you folks all know elephant? How were you able to figure that out? Pretty amazing, right? And you did that because your brain added information into what you were seeing. You had expectations. You gave meaning to the patterns of boxes. That's okay. Maya, most of you got it though. Pretty amazing how you can figure that out, right? And so that's what we would call the perception process. You giving meaning to the lines and shape that you saw, okay? The lines and the shapes are the sensation process. You understanding what you're looking at is the perception process. Pretty cool, huh, folks? Now, sensation and perception is actually one of the first areas of psychology. If you remember in chapter 1, we were talking about Gustav Fechner. I told you that he's probably the first psychologist, actually he wasn't even a psychologist, but the first person to record a generally accepted psychological measure. And I told you he measured the difference threshold, which was the amount that something has to change, a sensory experience has to change before you can tell that sensory experience has changed. So how much heavier does the weight have to get before you can tell you're lifting more weight? How much brighter does the dimmer switch have to get before you can tell that the room has become brighter? And so guys like Gustav Fechner measured our sensory experience in terms of absolute threshold, which was the absolute minimum amount of a substance that we can sense and perceive. And they also measured things like the difference threshold, which was the amount that a sensory experience has to change for us to understand that this change occurs. And what you're going to find is the subfield of psychology that's interested in sensation and perception is the experimental field of psychology. And they call them experimental psychology, not because they're the only ones to do experiments, but because they were the first ones to do experiments. And if you'll see right below me down here, you'll see some absolute thresholds to detect stimulus for each sense. So you can supposedly taste 1 teaspoon of sugar in 2 gallons of water, 1 drop of perfume in a 6-room apartment. Folks, let that be a lesson to you. There's no need to put on a lot of perfume. If we can smell 1 drop in 6 rooms, there's no need to bathe yourself in that cologne. We can smell you, okay? And so your experimental psychologists are interested in measuring these sort of sensory experiences. What are they going to do? They're actually helping us create brain bionic interfacing. I've got a friend, Sleeman Benzmaya. He works at the University of Chicago. He is super duper famous, and he is actually learning to program bionic fingers that you can actually move with your brain, that you can feel by your brain and move with your brain. And then there's also a branch of experimental psychology called signal detection theory. And these are the people that help us learn how can we diagnose tumors in your body. If I take a general picture, an MRI, how do I know that that little spot in your brain is really a tumor and not just some sort of weird aberration in your brain? Signal detection theory is the branch of psychology that's interested in helping us understand how to make judgments about whether or not we perceive something or not perceive it. And it has military and medical applications. Now let's talk briefly about the sensory cortices in the brain. Now we already tested on this for the first exam, but we're also going to test this again for the second exam. So I do want you to be familiar with all of the sensory cortices in your brain, okay? So you have a somatosensory cortex in your parietal lobe that feels all of your body parts. When you get touched on your shoulder or on your head or on your nose, your somatosensory cortex receives that sensory input, okay? When you taste something, right there in the back of your frontal lobe, you have a gustatory cortex. Right in front of that, you have an olfactory cortex for smell, okay? Your auditory cortex is located in your temporal lobe, Okay? And then your vision is processed by your occipital lobe. So we have all of these different areas in our brain that are responsible for receiving input from these sensory and receptor neurons that we have. Okay, now it turns out that you have a lot of areas in your brain that don't really seem to have a specific function. And one of the things that we have in our brain are called association cortexes. And these are where our brains put together the basic sensory experiences that we have so that we can perceive the unified whole. So when you're looking at Chris Roddenberry, part of your brain is perceiving my lines and my shape. Part of your brain is perceiving the color of my face. Part of you is perceiving the location of where I am. All that information has to get put together before you can come to a final perceptive conclusion about what it is you are looking for. And association cortexes are areas in the brain where your senses integrate together so that you can come to the perception of what it is you are looking at. Okay, now I do want you to know that all of your sensory experiences are processed through the thalamus except for smell. If you remember a couple of weeks ago, we were talking about the thalamus as being implicated in Tourette syndrome and a lot of other muscular problems like dystonia and Parkinson's disease. Well, the thalamus is also responsible for allowing you to consciously perceive most of your senses except for smell. Okay, so I do want you to know that about the thalamus for the exam. Now let's talk a little bit about vision. All right, so you have a picture in your book that's going to show you the different parts of the visual system. Things I want you to be familiar with for the exam. I want you to know the receptor neurons that are located in your eyes. You have these cells in the back of your eyeball, in your retina, called rods and cones. And these receptor cells turn light energy into patterns of action potentials. All right? If you'll remember last week when we were talking about consciousness, I told you that the left field of vision, your left hemifield is perceived in the right side of your brain, and your right hemifield is perceived in the left side of your brain. Now, each of your eyes views a hemifield. So your left eye has a left hemifield and a right hemifield image. Your right eye has the same left hemifield and right hemifield image. Now, those two images have to be transferred to the correct parts of your brain. And so we have this structure halfway back in your right in front of your thalamus called the optic chiasm. And that's where information from each field of vision is transferred over. So the things in your left hemifield from both your right and left eye can go to your right brain. And the right hemifield images that both your left and right eye see can go to the left side of your brain, to your left occipital cortex, okay? I want you to know that particular brain structure. Can you say what sense each part of the brain does, like sensory cortex? Yes, so the auditory cortex is for sound, okay? The gustatory cortex is for taste. The olfactory cortex is for smell. Okay, the somatosensory cortex is for touch, and the occipital cortex is for vision. Does that help, Riley? Okay, now there's one other structure I want you to be familiar with. It's called the superior colliculus, and it's located in the back of your thalamus. It's these two little green bumps down here that I have on the left picture, way down there. You'll see right there on the back of your thalamus. And the superior colliculus allows you to integrate your senses. So when I hear something, I know that I have to look this way. That's because my ears are tied to my eyes. I see a pen here on the desk in front of me. You folks don't see it, but I see it. And my eyes guide my hands down to that pen so I can pick it up. So the superior colliculus is responsible for tying all of your senses together. Yes, absolutely, Tara. Absolutely. So Tara Solomon was that in the video a couple of weeks with the Anil Seth. Absolutely. Your eyes are looking at your hands, and when that guy comes and smashes the rubber hammer with the hammer on the fake rubber hand that they've been rubbing with the little paintbrush, it kind of freaks you out. And that's because these two senses are tied together. And for a brief moment, you actually think the hammer is smashing that rubber hand, and it's your rubber hand. Absolutely, Tara. That's a very good thing to point out. Now, what I want you to know about the rods and cones. These are two different kinds of vision cells, and they have these things called opsins in them. And opsins are just light-sensitive proteins. The major one that you have in your eye that gives you vision is called rhodopsin, okay? And basically what happens when this protein gets stimulated, it causes a cascade of reactions to occur, which leads to an action potential that is then transmitted along the axon, the optic nerve, which is just the long axon that goes from your eyeballs all the way to your back of the brain, and it transmits this action potential from the eyeballs all the way to the back part of your brain. Now, you've got two different kinds of receptor cells. You've got rod cells, which work very well in low light. If you've ever turned off the lights at night and waited a few minutes, you can see shapes all around you in your house, right? You don't bump into things because you have pretty good night vision. I'll answer that question in just a minute, Etta. That's a very good question. Okay, and so these rods cells, they work very good in low light, and they give us sort of our night vision, if you will. Now, they're located out of the fovea. What is the fovea, you ask, Chris? If you are looking directly at something, that image is falling on the middle of your retina. The middle of your retina is called the fovea. That is where stuff is at when you are looking directly at it. Your rods and cone cells mainly just see outside of the fovea in your peripheral vision. Now, if you look directly at something, that image is falling on your cone cells that are located mainly in the fovea of your retina. Now, cone cells need a lot of light, but they help us generate color vision. You folks may not believe this, but you really only have color vision in the middle of your field of vision. If you think you see color around the periphery of your vision, that's your brain adding in color information that it expects to see. You are not actually seeing color, but your brain is adding in color around the periphery of your vision. If you'll go back, all of this is going to relate to Anil Seth's talk. If you go back and look at his video, he talks about how we use expectations to make judgments about what it is we are seeing, we are hearing. If you'll remember, we had I think Brexit is a very bad idea. If you remember that, you couldn't understand what he said first time when he said it in that weird computer voice until he said it the right way, and then you were able to hear it in the computer voice. And so your brain sort of added in information which allowed you to understand what he was saying with that computer voice. You do the same thing with color vision. So I'm looking straight ahead. My rods and cones are seeing color vision right here, but then my brain is adding in color all around the periphery of my visual field. Okay, now Etta asks a question. I've got the video this week for you to watch about the blind boy who learned how to see with his ears, and he echolocated. Now the weird thing about him is he actually was receiving sound information through a different structure. His ears. And what happened to this gentleman is probably the neural connections from his auditory cortex invaded his visual cortex because he didn't have eyes. So he sort of had this area in his visual cortex which didn't have anything to do. And so as he starts hearing, those two brain areas sort of knit together. Remember, we talked about neuroplasticity as a way for your brain to overcome brain damage. When he lost his eyes, he had some area in his occipital lobe that didn't have anything to do. So this part of the brain starts looking for something to do, and it knitted in with his auditory cortex. So he was getting information that was coming into his ears, but it was going to his visual cortex as well as his auditory cortex. And he learned how to interpret that as vision, if you will. I hope that makes sense, Etta. Okay, so you have these rod cells and these cone cells located in your retina. They are sensitive to light. When they experience light or changes in light, they generate action potentials that travel along the optic nerve. And remember, they go to your thalamus. Remember, some of them go to your suprachiasmatic nucleus to give you, to help you stay aligned with your light and dark circle. Some of them go to your superior colliculus, which allow you to coordinate your vision with your other senses. All right. And then the majority of these nerve fibers, about 90% of them, go back to your occipital lobe to give you what we call conscious vision. Now, I want to talk briefly about color vision. It turns out that you have 9 different kinds of opsons in your brain. 9 different kinds of opsons. Now, the one that really does most of the work and generates what you might call your conscious vision is rhodopsin. It's a light-sensitive protein in the eye, and it causes action potentials to be generated when it's stimulated by light. Now, your cone cells actually have 3 different kinds of opsons, and each of these opsons is sensitive to a different part of the visible light spectrum. So if you look at this picture right here, this picture represents the visual light spectrum. This is all of the light that can be perceived by your rods and cones. Now, I don't know if any of you have ever watched superhero films, but if you've ever seen Superman, Superman has X-ray vision. Now, X-rays are nothing more than a light waveform that falls out of what we would call the visual light spectrum. So X-rays are bouncing around, but your eyeball is not sensitive to light in that wavelength. Instead, your eyeballs are sensitive to light. They respond to light that falls somewhere between 400 nanometers and 700 nanometers in length. So you see this waves, how they have a top and a bottom. If you go from top of one wave to top of the other wave, that's what we call the wavelength. All right. And it turns out that you have 3 different cone cells that respond to different parts of the visible light spectrum. So you have blue cones. They only respond to light in the shortest wavelength. Good question, Kaylin. We're gonna talk about that in just a second. You have green cones, and then you have red cones that respond to light in the longer wavelengths. It turns out you have the cones. All of the cones. What probably happens, Keelan, is your genetic instructions don't make the protein that responds to that particular color. So the cone is still there, but you might be missing the red opsin, the opsin that responds to red, or you might be missing the opsin that responds to green. And so most of the time, people who have color blindness, the most common kind is what we call red-green color blindness. And so they don't have the 2 different opsins that allow them to distinguish the red color from the green color. Does that make sense? Okay, now, bizarrely enough, It absolutely does, Riley. It works just like an RBG color scheme in that we engage in this color mixing. Now, there's also this process. We're not going to talk about it for the exam, but it's called the opponent process. It turns out whenever your red cone is firing, not only is it firing, but what it does is it inhibits the firing of your blue cone. Okay, so while your red cone is active, it actually makes your blue cone inhibited, right? So we have kind of this opponent process going. If you've ever done that experiment where you look at a black and white image for 30 seconds, right? And then look away to a white wall and you see the opposite of the image you were looking at. Has anybody ever seen that demonstration before? The opponent process. What happens is when you're looking at one color, the cone cell that sees that color is actually inhibiting the other color so that they can't see. So not only do we have these three different colors that mix together, but the colors also work by an opponent process so that when you see one color, the other color is impaired. It shows the negative version. Exactly. And what happens when we see the negative version is as soon as the red cone lets up, the blue cone doesn't come back to normal. It actually bounces back like a spring and gives you a brief vision of whatever color was being inhibited at the time, right? Does that make sense? And so we have these two processes, sort of the trichromatic process going on here with three different cone cells, but we also have an opponent process, and it's controlled by these cells. If you go back to this picture right here, there are these cells called ganglion and bipolar cells. If you see them in the chart right here, and the bipolar cells, when the red cone is firing, it tells the bipolar cell, hey, I'm firing, and the bipolar cells, well, as my red cone is firing, then I'm going to inhibit the green cone so it won't fire. And so that's why we don't see reddy green, you know what I mean? You either see red or green because that opponent process is working. I hope that makes sense. Now, so here's the deal. In your occipital cortex, you have 5 different areas that engage in primary processing. So you have one part of your brain that looks at lines and the direction that those lines go. The other one looks at sort of the location of those lines, so movement, then also the location of the line. You have a part of your brain which is responsible for color vision as well. So you have these different areas that sort of build up the basic parts of the visual world. So you do what we would call primary visual processing, where you process size, angle, and movement and location. And so these are all what we would call the primary visual input. And you actually have a part in your occipital cortex called the primary visual processing center. And that's where lines, colors, and shapes are processed. Then that information is moved along to a visual association cortex, which then begins to put those things together and come to an idea about what kind of shape it's looking at. That information is then sent to another association cortex, which says, you know what, I've seen that shape before, and that's probably Chris Roddenberry, is that shape that I'm looking at. Now, sensation is seeing the shape, the line, and the color. Perception, on the other hand, is being able to figure out what it is you are looking at. And that's what we would call the process of perception. Now, your book talks about 2 general processes of perception that visual researchers are familiar with. And the first represents the way we organize our visual information. All right, so one way we can see the world is by using what we call bottom-up processing. You can see the lines, you can see the shape, the color, and the movement, and you can put everything together and say, what is this thing I'm looking at? And you can use the parts to come to a decision about what the whole is. And that's what we'll call bottom-up processing. But human beings also use guesses to figure out what they're looking at. So you folks only see half of me right now. But I'll bet you probably know that I have legs too, even though you've never actually seen my legs, correct? Okay, we'll call that top-down processing because you saw part of me and then you added in the rest of the image to come to an idea about what you're looking at. Yes, you do. Did anyone else freeze up? I don't think anybody should be frozen. I'm looking at the feed right now and it looks like we're going. I'm not sure. Okay, yeah, it might be just you, Camilla. You have legs. Yeah. Look at that. Look at that. You can see a leg in a boot. All right. Okay, cool. And so that's what we would call top-down processing. Now, it turns out that human beings, over the course of the first couple of years of your life, you learn these rules. You learn these rules that help you make guesses about what it is you're looking at. Remember that guy Anil Seth talked about how really what we're doing is we're making guesses about what it is we're looking at. He was talking about top-down processing. And so human beings learn these patterns of perceiving the world so that we can make guesses about what it is we're looking at. And so your book talks about the figure ground principle. One thing that we typically do is we learn how to look at objects and separate them from the background. So you'll see, you probably guess that you're looking at Dr. Roddenberry floating in front of a blue background. Do you notice how you separated that out? And so you've got Dr. Roddenberry in front, and you know that the blue is the background. And so you have learned how to separate out the figure from the ground. That's one of the first rules that we learn. Some objects are the figure and some things are the ground. But not only we do that, we learn that our world tends to occur in certain patterns, and we learn to use these patterns as guesses when we perceive the world. And these patterns were first identified by these psychologists, these German psychologists in the 1930s, who thought that really if we wanted to understand vision and perception, we needed to look at the whole Gestalt, or the whole thing. Human beings, although we perceive the individual details, we always put them together so that our conscious experience is a whole and not individual pieces. This is what we will call the Gestalt way of perceiving things. And these Gestalt psychologists figured out, I am, aren't I? That's my history as a military guy. I'm just kidding. I was never in the Army. I don't want you to think that. Not because I think the Army's bad, but because I didn't earn it, because I didn't go to boot camp. If you didn't suffer through boot camp, don't joke about being in the Army. Ha ha. Where am I? Now you can't see me. Okay, so these Gestalt principles of grouping are just ways in which you learn how to perceive the world. So, for example, if you look here on the far left, you do not see 15 blue dots. Most of you probably see 3 groupings of dots. So what we tend to do is we tend to put things into groups based on how close they are to one another. This is called the Gestalt principle of proximity. If I asked you what you were seeing, most of you wouldn't say, I see 15 dots. Most of you would say, I see 3 groups of dots. All right. Now, in the second picture here, if I asked you what you were seeing, you probably wouldn't tell me, I see a bunch of lines. Instead, you would probably see, I see some squiggly lines and I see some straight lines. If you say, I see some squiggly lines and some straight lines, then you have organized that image based upon similarity. If you were never through basic training, you're not worthy of joking about the military. Absolutely. So don't come beat me up, Riley. Okay, I take that back already. Please don't get me. Okay, so that's the second Gestalt principle. The third one is continuity. Most of the time, we can tell, because we understand figure and ground, we tell that some objects are in front of and behind others. So if you're looking at picture C, you're not going to tell me I see a pink cylinder with a green bar on the left and a green bar on the right. What you're going to do is continue that green bar behind the cylinder, and you're going to say, Chris, what I see is a long green bar behind a cylinder. So what you have done is you have continued that image. Even though you can't really see it, you added it in your mind. Okay, picture number 4. What do you see in picture number 4? Can somebody tell me? What do you see in picture number 4? I'm going to ask you real quick. Does anybody tell me what picture 4 is? A triangle. Okay, Etta, you are wrong. Keelan, you are wrong. Tara, you are wrong. And Nicole's only sort of wrong. Actually, there are 3 angles there, right? I'm just kidding. What did you do? What did you do? You actually finished in the sides and turned that into a triangle. It wasn't really a triangle, but you and I and everybody in this class knows that that's supposed to be the shape of a triangle. Now, if you think about it, this is kind of deep. If you think about it, that's not really a triangle. You have to add something in your mind before you can see a triangle. It's not just there. You have to add that information in. And if you look at number 5, instead of seeing 4 Pac-Mans, 4 little semicircles with pizza shapes cut out of them, you probably see a box that is outlined by 4 circles, right? And so that's what we would call illusory contours. So these 5 ways, proximity, similarity, continuity, closure, and illusory contours, these are ways in which we begin to make guesses, like Anil Seth said, about what it is we are looking at. The first guesses we make are based on these Gestalt principles. On your exam, I'm going to show you some Gestalt principles, and I want you to be able to identify them. What you should do today when you go out and walk around your world is see if you can identify places where you make guesses and use top-down processing to make guesses about proximity, similarity, continuity, closure, and illusory contours. It does kind of look like a flower, Etta, now that you mention it. Okay, now, here's the thing. Human beings also use guesses and top-down processing for vision. Now, if you look over here right underneath me, you will see what's a term called binocular disparity. Remember, I told you your left eye sees the left world and the right world, and your right eye sees the left world and the right world. So it turns out that you're getting 2 different pictures of the left side of your world and 2 different pictures of the right side of your world. What happens when that information gets all the way to the back of your brain looks at the differences in those 2 images because they're both taken from different places. This eye's over here and this eye's over here. And your brain compares the differences in those images and gives you an estimate for how far that object is away from you. This is what we call binocular disparity. And if you want to see binocular disparity, just close one eye and bounce your eyes back and forth, left to right, and move your finger towards you. And what you're going to notice is as your finger comes closer towards you, when you close and open each of your eyes, your finger's going to bounce left and right, left and right. And that's because each eye is seeing that finger from a slightly different direction. This binocular disparity gives us what we would call true depth perception, the kind of depth perception that allows you to play baseball. Okay? Now, it turns out that human beings also learn some ways to guess about depth perception based upon how the world is organized. And we're going to call these guesses that you make about depth perception, we're going to call them monocular depth cues. So I have a picture right here in front of you, and even though this picture is flat, I guarantee you can tell that that dude in the tie is in front of the car. I'll bet you can tell me that those ladies walking across the crosswalk are closer to you than the ladies standing on the sidewalk behind them at the stoplight. How do we make these judgments about depth, even though there really isn't true 3D depth in this picture? These are called monocular depth cues. So if you could, what I'd like to do is draw you a picture and demonstrate some of these binocular depth cues to you. Now I want you folks to watch carefully, because this is going to be some of the finest art that you have ever seen in your entire life. Let's go to the whiteboard. Let's erase the elephant here and the dog and the cat, and watch this. Boom. This is a picture I'm very good at drawing. It's my two mountain picture. Mountain A and mountain B. Which mountain is in front, A or B? If you say A, absolutely. And this is called this is a monocular cue we're going to call occlusion. A is in front of or occludes B. And so that's one way in which we guess about height. Now, you know what? I'm going to do this right here and this right here. I'm going to draw a road. Now, this represents two cues. One of the things we know about parallel lines is that if they go away from you, they tend to go away and meet at a point. You see how the lines of this road meet way off in the distance? Now, we as human beings know that that is not exactly what happens, but your brain interprets this as distance, and we're going to call this the linear perspective, okay? Parallel lines tend to meet off in the distance. Now, do you also notice the lines in the road? Do you notice how the lines are far apart here, but as we move away, they start to get closer? This is what we call the monocular cue of the texture gradient. So as we get farther away, designs start to get a little bit closer together. This is what we call the texture gradient. Now I'm going to put a couple of this is a desert picture that I'm drawing, and there's a cactus, and then I'm going to put a cactus here. Which cactus is closer to me? Cactus A or Cactus B? Cactus A or Cactus B? It is Death Valley. You're right. Which one's closer? Cactus A. Why is it closer? Because it's lower in your field of vision. It's lower in your field of vision. So height in the field is another monocular cue that we learn to use in order to make judgments about what things are. Now, here's the other thing too. Let's see. There's dude. That's Fred. That's Fred. And this is Ted. Which one is closer to you, A or B? Which dude is closer to you, Fred or Ted? Absolutely, Ted is. And that's because, here's the deal. We know that people are about the same size. There is no way that Fred is teeny tiny compared to Fred. So we know that these guys should be the same size. So relative size and familiar size allows us to make judgments about how far something is off in the distance. Do you see this picture? Is this not the most beautiful art picture that you have ever seen in your entire life? Okay? And so, in a sense, this is how we perceive vision. So here's the upshot from today's lecture. Okay. Now, human beings engage in two processes. We sense things, we sense smells, we sense sounds, we sense sights. And then when they get to the back of our brain, based on our experience, we learn how to make guesses about what these sensory experiences are. And we call that perception. Right? And we have these very special neurons called receptor neurons that transduce physical energy into patterns of electrical impulses or action potentials that we then sense and then turn into perceptions. The one part of your brain that's responsible for vision is the occipital lobe. We have rods and cones that perceive light. Rods are good for giving you shapes and really good black and white vision in low light. And your rods and cones help give you color vision. Information from your rods and cones in your retina goes all the way back to your occipital lobe. It transfers lobe at the optic chiasm right in front of the thalamus. And if you remember, some of that information goes to your suprachiasmatic nucleus to keep you tied to the sun. Some of that information goes to your superior colliculus to tie your eyes to the rest of your senses. And most of that goes to your primary visual cortex, which processes vision. Now, not only do we process the basic elements of vision, we'll call that sensation, but we also make guesses based on our experience as to what we see. And there are two sort of perceptive processes that are going on. The first is represented by the Gestalt principles of vision, where we organize things into groups and sizes and shapes based upon our prior knowledge. And then there are also what we call monocular depth cues, which help us perceive depth, even when we might not be able to see it accurately with our two eyeballs. But what happens if my retina gets ruptured? Then you, my friend, will have difficulty seeing. Absolutely. Absolutely. You do have the modern Michelangelo. And by the way, I will be selling this picture after class for the very low price of $12,000. If anybody wants to bring their credit card after class, I'd be more than happy to sell this. And you know what, doggone it? You know what? I'll even throw in an autograph for you. Look at that. I'll even autograph this picture for you. How about that, right? Okay, folks, thank you very much for coming. I know Jack will do 100,000. Sweet. Thank you very much for your time. I'm sorry to keep you 50 minutes today. I hope you learned something out of today's lecture. Take care. I look forward to seeing you on Thursday. Oh, by the way, we've got a webinar tonight. Please watch the video, The Blind Boy Who Can See. Please watch the video, The Blind Boy Who Can See, before you come to class tonight. It's going to be an absolutely amazing webinar, and I look forward to seeing you at 8 o'clock tonight. Does anybody have any questions? If you don't have any questions, I'm going to say goodbye. Take care, and I'll see you next week.